And joining us at the University of Colorado Cancer Center and Cancer Biology PhD program symposium series. I'm Claire Isle. I'm a grad student in the Cancer Biology program, and I invited Dr. Rachelle Johnson to share her work with us today. So Dr. Johnson is an assistant professor of medicine at the Division of Clinical Pharmacology at Vanderbilt University Medical Center, as well as the Ingram Cancer Center linked to Vanderbilt University. Dr. Johnson received her PhD in cancer biology at Vanderbilt in the labs of Gregory Mundy and Julie Sterling, then continued on to her postdoc with Dr. Natalie Sims and Dr. Jack Martin at the St. Vincent's Institute of Medical Research in Melbourne, Australia. So that's a big move from Tennessee. And then she returned to the US to also do a postdoc in Dr. Amato Giacchia's lab at Stanford University. So since then, she's returned to Vanderbilt, and really her research has been focused on mechanisms driving tumor cell dormancy and processes supporting tumor cell colonization to the bone. So thank you all for joining us, and thank you, Dr. Johnson, for joining us today to share your work, and we're excited to hear what you have in store for us.
Here, let me put my slides. Okay. Can you see it okay, Claire? Oh, hold on.
Yeah, we see your, there you go, there's presenter view.
Perfect, let me put that up here. Great. So much for the introduction and for the invitation to present today. So I'm planning to cover two different projects in the lab right now, and these are both unpublished. One was very recently submitted and are still, I think, very much works in progress, even for the one that we're trying to wrap up right now to distant sites. Check the chat. Great. I'm going to make sure it wasn't an issue. So I will just dive right in, and I think that most folks are familiar with the general process of tumor cells leaving the primary tumor site, but we know that disseminated tumor cells, when they actually leave the primary tumor, that there are multiple fates for these cells. So after intravasation and survival in the bloodstream and extravasation in a distant organ, cells can undergo cell death. They can either become dormant, where they may exist as a truly quiescent G0, G1 arrested tumor cell. They can become a dormant micrometastasis, where they really don't expand and become clinically detectable. A really aggressive metastasis within any distant organ site. So one of the things that we also understand is that this process of dissemination actually happens much earlier in tumor progression even than we thought. So for example, patients who have ductal carcinoma in situ, even those patients can have disseminated tumor cells in the bone marrow well before you even have detection of like a full primary tumor. So the issue with this is that it's these disseminated tumor cells and ones that are give rise to recurrence. So what does this look like within the bone? So when breast cancer cells metastasize to bone, and particularly once they sort of exit dormancy and become this really aggressive growing tumor in the bone, you can get really dramatic sort of quality of life issues that can occur for patients. So this is an example of a fracture of a patient who had a breast cancer recurrence within the bone. And so obviously these can really impact patient mobility as well as mortality. Look at just the incidence of bone metastasis, different tumor types. We see that up to 70 to 80% of breast cancer patients actually present with bone metastases upon autopsy. So we know that this is really a pretty I'd say, prevalent problem within the breast cancer population. So once tumor cells disseminate into the bone marrow, they encounter a lot of different signals. So they can firstly compete with hematopoietic stem cells or HSCs.
Listen, Doctor, your audio, sorry, this is James. Your audio is a bit unstable.
Oh, no.
you might want to try just turning your video camera off. I don't know if that'll give you better bandwidth, but I mean, I've been able to understand it all, but it is kind of breaking in and out.
Okay, give me just a second. I'm going to do that. I can get this pulled up. Okay, here we go. Let me stop video. Okay, let's see if this works. And if you're still having issues with it, then just pop up and let me know again. Okay.
Actually, it still seems to be a problem. I'm not sure what else. The only other thing is you may want to try just getting closer to the mic. I don't know. Are other people hearing the same thing I'm hearing? Is it just me?
Yeah, let me know. And I can't. Yes. Everyone else having issues as well?
Yeah, you're cutting out just a little bit. Maybe if you have headphones, could you try that to see if that will help stabilize the audio?
I do not. If it becomes a problem, let me know again, and I can also try changing locations. Okay.
Yeah, I think we can all understand it still. Sorry for the Zoom technical difficulties.
I know, right? Okay, thank you. Yeah, and definitely just if it's you know, if it really becomes a major issue. Okay, so breast cancer cells then, once they reach into the marrow, then as I mentioned, they will encounter lots of different signals. They will compete with these SCs for the bone metastatic niche. And these are just some of the different signaling pathways that have been identified over the last several years that can regulate tumor dormancy and tumor escape from dormancy within the bone marrow. So once tumor cells begin to grow and proliferate through these changes in signaling, then this is what really leads to this metastatic outgrowth. And what happens at this point is that tumor cells will secrete lots of different cytokines, like parathyroid hormone-related protein is one example, and that'll come up quite a bit in the second half of this talk. And PTHrP will actually induce osteoclastogenesis through osteoblasts within the and this will release lots of growth factors and cytokines from the bone matrix. And this actually feeds back onto the tumor cells to promote their growth and proliferation and the bone destruction that occurs. So, so PTHrP in particular is one of those key molecules required for tumor-induced bone destruction. So, so there are obviously lots of different signals that the tumor cells will encounter within the bone marrow. So they'll receive these signaling, you know, these growth factor cytokines. They will also encounter things like the matrix stiffness, so they can encounter different rigidities, and then they will also experience hypoxia. I just saw in the chat well that maybe try with the phone, to try calling in with the phone. So hold on.
I think that might be a good idea. Do you need me to send you the link again?
I don't think so, as long as it'll let me join from my email from my phone. So give me just a second. I will try that.
Sure. Thank you.
Mm-hmm. Oh. Now I can't really hear anyone who speaks up very well. So if you cut in, then just, you know, be plenty loud. So okay, perfect. I will keep going. All right, let me X out of the chat. Okay, so so the tumor cells will in addition to all of those mechanical cues, as well as the signaling cues, they will also encounter hypoxia within the bone, because the bone is a physiologically hypoxic organ. So tumor cells will also encounter pretty different oxygen levels depending on where they're at. So within the primary tumor, you can have tumor cells that can have variable oxygen levels that they encounter due to the disorganized vasculature that forms within the tumors. And then when they disseminate to distant sites like the lung in particular, they'll encounter pretty high oxygen levels, anywhere from 10 to 20%. But once they disseminate to the bone marrow, then it's typically going to be somewhere around anywhere from less than 1% up to about 9%, depending on exactly where the cells are within the bone microarchitecture. So one thing that's important to keep in mind, though, is that in all of these sites, that the tumor cells can experience hypoxia. So, for example, you know, the tumor cells in the primary tumor, as I mentioned, they have this disorganized vasculature, and this can then lead to, you know, pockets of hypoxia within the primary tumor. Within the lung as well, even in this really super, you know, super oxygen, you know, levels that are available, you can still get hypoxic regions within the tumor cells that grow out in the lung. And then of course, within the bone where it's already physiologically hypoxic, you can get tumor cells that experience hypoxia. And this is all pibinidazole staining within these sections here. So one of the main interests that we've had in the lab is, first, how does hypoxia in that primary tumor site actually impact dissemination to distant sites? And then also, you know, the outgrowth of the cells in those distant sites as well, which is what I'll talk about a little bit more later. So for those of you who are not familiar with hypoxia, in normal oxygen levels that cells encounter, you have factors that are called HIF1 alpha and HIF2 alpha, as well as HIF1 beta. And these factors can be degraded by the proteasome, and they're targeted by VHL, which is an E3 ubiquitin ligase. That will lead to ubiquitination of HIF1 alpha and HIF2 alpha. But in hypoxia, the enzymes that actually hydroxylate HIF1 alpha and HIF2 alpha, which are the PHDs, that these enzymes are inhibited, and this allows the HIF factors to actually translocate into the nucleus and induce lots of gene transcription and activation of many different signaling pathways. So in the context of metastasis, and in particularly to the bone as well, there's quite a bit that's been published over the years, especially in roughly the last 20 years or so. So one of the most important things is that we understand that from the clinical data that patients who have elevated HIF1 alpha staining in breast cancer patient samples, that those patients tend to have a poorer prognosis. And then we also know that it's been previously shown using a genetic model, which is actually the same model that I'm going to present here that we used, that if you delete HIF1 alpha in the primary tumor, that this will actually reduce lung metastasis. And then it's been shown in the context of bone metastasis and bone colonization that if you overexpress HIF1 alpha in metastatic breast cancer cells, that you can increase their ability to colonize the bone. And this can happen whether you do an intracardiac injection, which bypasses some of those early steps of metastasis, or orthotopic inoculation, which is a little closer to the full you know, metastatic cascade. And then also they've shown that knockdown of HIF1 alpha, so sort of the converse experiments, will reduce bone colonization. So what we had been interested in looking at, though, was to try and evaluate the impact of HIF1 alpha in the primary tumor on dissemination to bone as well as to lung. So trying to look simultaneously at what's happening in these different sites and using that spontaneous genetic model. So essentially doing what sort of was presented previously to look at lung metastasis, but now we really wanted to focus on bone. So this was a project that was started by my student Vera, who will, I think, be finishing up sometime in the next few months. And what she did was to generate a mouse model. So this was sort of a triple mouse model where she crossed MMTV Cree mice that will express Cree specifically within the mammary fat pad, and crossed these mice with HIF1 alpha flox mice, and then crossed those mice with polyomidal T mice that will spontaneously develop mouse mammary carcinomas. So what she did then is to collect
mainly, so just for the nomenclature, so it's clear for the rest of this, she collected HIF-1 alpha flox mice is our terminology for them. And these mice are basically our wild type mice. So they were wild type for the Cree, and then had the HIF-1 alpha flox and the polyomidal T transgene. And our HIF-1 alpha knockout mice were positive for the Cree transgene. And you'll see in some parts of the data as well that we've also included the polyomidal T negative controls, particularly for the bone analysis. So what she did was to monitor these mice for spontaneous tumor generation. That takes roughly about 8 weeks. And then it takes about another 12 weeks for them to then reach our euthanasia criteria, which was for the tumors or one tumor at least to reach 1 centimeter in diameter. So the polyomidal T mice will develop, some mice will develop multiple tumors. And so we were just watching for that, for the first tumor to reach 1 centimeter, and that's when they were sacrificed. So her hypothesis was that deletion of HIF-1 alpha would reduce tumor dissemination to distant metastatic sites. And that really was based on what we had seen from the previously published lung metastasis data using this same model. So the first thing we noticed was that the HIF-1 alpha knockout mice actually took a bit longer to reach the euthanasia criteria. But upon sacrifice, those mice actually had slightly higher total tumor burden. So even though we were monitoring that one tumor to hit 1 centimeter, in diameter, by that point, in general, the total tumors were larger overall within the mice. And this was maybe partially driven by the number of tumors. This was not a significant increase, but I think it could have bumped this up a little bit. So it's possible that that sort of helped it, but it seems to be mostly driven by the tumor size. And we did also confirm for this and for the other mouse models that I show later, a significant reduction HIF-1 alpha at the expression level. And then we also looked at, we verified by recombination as well that we had deleted the exon that we intended. So the first thing that we did then was to look at the dissemination of the tumor cells to the bone in these mice. So the best way to do this was using some techniques that we had published on about a year previous, where we can use either flow cytometry of flushed marrow from the bones and sort for EpCAM positive tumor cells, or we can actually homogenize the bone samples and extract for RNA and then do qPCR on one of the genes within the cell. So in this case, it would be the polyomidal T cells. So we first used from the femur and tibia. We took the marrow from those mice, and we saw that there was a significant reduction in dissemination of the tumor cells to the bone. And we saw a trend in this direction with the spine, but it wasn't statistically significant, but fit with what we had seen in the femur and tibia. And so when we looked at the histology for these mice, we were curious to see whether we would actually be able to detect at the histological level the tumors in the HIF-1 alpha just wild type mice with the polyomidal T transgene. And we really can't. So we didn't detect any mice with tumors by histology. So they weren't really like taking hold in the bone marrow. And so naturally we couldn't detect any kind of decrease here with the in the HIF-1 alpha knockouts at the histological level. But we did also perform micro CT on the bones from these mice. And we did this because you have to run the micro CT before you can process for histology. So we had already done the micro CT for these mice, and we went back to analyze. And so we had expected to either see no change in bone volume, or if anything, we had thought before we really looked at the histology, we thought we could potentially see an increase in bone volume because we had less tumor burden in the bone, and knowing that these cells can become bone destructive. So we were surprised to see, though, that the bone volume was actually significantly lower in the HIF-1 alpha knockout mice. And this was quantified by micro CT. We haven't quantified the histology for that either, but it does seem to be evident as well within the histology that we have slightly less bone there. So we are not sure why we have less bone volume there, but this is one of those areas where we did include the controls that did not express the polyomidal T transgene. And what's interesting is those mice do not have any difference in bone volume between each other. So to us, this suggested that it's likely not an off-target effect of HIF-1 alpha deletion in a different cell type. Otherwise, I would have expected to see the same thing here. So it suggests it's something due to the presence of the polyomidal T tumor cells, but we're not really quite sure how that's happening, because we clearly have pretty few tumor cells that are present in the bone marrow. So that's something that we are still trying to sort out. So the next thing that we did was to look at the lungs, and this was really, we approached this part of the experiment as just sort of an extra control, because it had been previously published that these mice have a significant reduction in lung metastasis. And so we were actually pretty shocked to find that we had a significant increase in the number of metastatic lesions, and that we also had a significant increase in the total metastatic lesion area and an increase in the incidence of lung metastases. So the number of mice who actually developed pulmonary lesions was higher in the HIF-1 alpha knockout group. So I should note here as well that all of this work with the lung data was performed by our board certified veterinary pathologists as well, that we had handed these samples off to them. And so they had scored all of this. And so we still don't really understand why there's a discrepancy with what was previously published. But it made us wonder whether HIF-1 alpha is maybe essential for the dormancy induction in lung disseminated tumor cells. So this would actually fit with data that's been previously published by Julio Aguirre-Guizzo's group, who's really led a lot of the work in the dormancy field, where they've shown that HIF can actually stimulate dormancy within breast cancer cells and with other cell types as well. And so we wondered, maybe these data are really consistent with that idea. So we wondered if we would see changes in dormancy markers if we looked at the tumor cells, either in the primary tumor or in the lung metastatic site. So, Vera then embarked on quite a journey to try and find some sort of mechanism for this effect. It's the first thing that she tried was to look in the lung metastatic nodules at proliferation, baseline proliferation. Do we see a change in Ki-67? And she did not see any difference in proliferation for those cells. So we had thought perhaps we'd see an increase in the HIF-1 alpha knockouts, and that did not happen. So she also examined whether or not we had differences in CD4 and CD8 tumor cells within the lungs. So we thought perhaps this could be mediated by immune clearance. Maybe we have less clearance of the tumor cells within the lungs, but we also saw no significant differences in the immune cell markers. We also looked within the primary tumors as well to just see were there any differences in some of the other sort of low-hanging fruit for dormancy. So autophagy, dormant cells have been shown to upregulate autophagy, and we didn't see any difference between the wild type or the knockouts. That was significant, and the same for an EMT panel as well. And so I think, at this point, Vera was really frustrated and trying to work out what was going on. And so we decided to sort of leave that where it was at the time and let her go on to characterizing some of the other mouse models that she had crossed. So in addition to the HIF-1 alpha knockout mice, she had created 2 more transgenic lines where she had deleted HIF-2 alpha or had deleted VHL, which would increase the HIF signaling. So she decided for both of those models to go in and go ahead and look at the lung and bone dissemination and see if this would help reveal, some potential mechanisms for the HIF-1 data. So what she found was that if you just look at lung dissemination, there's no difference in the lung homing in the HIF-2 alpha knockout mice. So this is either in the number of metastatic lesions or the total lesion area. It was unchanged with HIF-2 alpha knockout in the primary tumor. And similarly, there was no difference if she knocked out VHL. So we again saw no difference in the lesion number or lesion area. When she looked in the bone for these mice, though, she saw that the HIF-2 alpha knockout mice had a significant reduction in dissemination to the bone, just like the HIF-1 alpha knockout mice had. And the VHL knockout mice also had a sort of converse increase in dissemination to bone. So this really fit with sort of the original hypothesis, which was that we had expected we might have a reduction in bone dissemination with HIF1 knockout. For HIF2, it was sort of valid as well, because it's previously been shown that HIF2 alpha can regulate PTHrP. And so we thought, potentially that we could see effects on bone metastasis there as well. And for VHL, obviously, we had assumed that if we elevated HIF1 or HIF2, that we might see that increase in dissemination. But when we looked at the bone volume for these mice, so we did the same analysis that we did for the HIF1 alpha knockouts, we saw that there was no significant change in bone volume in these mice, and same for the VHL knockout mice. So this was a little bit different from what we saw with the HIF1 knockout mice. So those mice were the only ones that had a reduction in bone volume. So this sort of summarizes where we were at, where the HIF1 alpha knockout mice have this decreased bone volume, as well as a reduction in bone dissemination, but then an increase in lung dissemination. And so it struck us at that point that we did not see any changes in sort of the bone metastatic niche in the HIF2 alpha or the VHL knockout mice. And so this sort of led us toward this thinking that perhaps what's happening is that we are changing sort of the potential for these tumor cells to engraft within the lung by reducing the available osteogenic niche where these tumor cells could engraft in the bone and even potentially stay dormant there as well. Due to the signals from really from both the osteogenic niche as well as potentially even the perivascular niche in the bone. At this point, that still remains sort of our working hypothesis here. And we are doing some follow-up studies right now to see, look at different markers within the osteogenic niche to see if those are reduced within the bone, within the HIF1 alpha knockouts. But we also still needed to validate or try to track down some sort of mechanism for the HIF2 alpha and VHL effects, at least on the bone dissemination. And so as I mentioned before, HIF2 alpha can regulate PTHrP. And so we decided to look at PTHrP expression levels within the tumors of the wild type and the knockout mice for each of our 3 strains. So the VHL knockout mice here, you can see that we saw a slight trend towards an increase in PTHrP, really no change with HIF1 alpha, and then a trend towards a reduction in HIF2 alpha. So although these weren't significant, it fit with sort of the general idea that HIF2 alpha, I think, can stimulate PTHrP. And so it suggests that it could maybe be a potential mechanism. At least the data, I think, are somewhat consistent with that, but certainly still far from, I think, really showing that that's mediated through PTHrP at this point. So we still have more work to do to sort that out. But we also then looked at CXCR4 in terms of trying to look at the bone homing. And we were really trying to think at this point too about things that may be more specific for bone versus the lung. And so when we looked at CXCR4 expression, we actually saw that there was a significant increase in the VHL knockout mice and no change in the HIF1 alpha or the HIF2 alpha knockout mice. And so our working hypothesis with all of this then and the potential mechanisms there is this. And it's summarized sort of in this table as well as the figure. So whichever is easier for you to follow, feel free to look at it. So within the HIF1 alpha knockout mice, again, we think that potentially the increase in lung dissemination could be due to this reduced osteogenic niche. And for the HIF2 alpha knockout mice, we think that the decrease in bone dissemination here is not because the osteogenic niche has been reduced, but rather because we may have a reduction in PTHrP. And there's almost certainly other mechanisms there as well that are at play. And for VHL, we think that one of the ways in which it could potentially be increasing bone dissemination is actually through an increase in CXCR4 signaling. And really kind of intriguingly, this seems to possibly be even independent of HIF1 and HIF2 alpha as well. And so that's where we've sort of landed with our mechanisms so far. But we are I think still have a good bit of work to do to try and nail those down. So, okay, so moving back to our idea of sort of hypoxia and looking at how hypoxia can impact the tumor cells in different sites, really the question that we have been very interested in is how the physiologically hypoxic microenvironment of the bone marrow actually impact dormant tumor cells. And that has actually been the toughest question, I think, to go after in terms of approach. So we actually have some engineering models that we are working with our collaborator, Dr. Scott Gelcher at Vanderbilt, to try and do, I think, some more pointed experiments to really get at how hypoxia within sort of a fabricated osteogenic niche can actually regulate tumor cell homing and engraftment into that niche. And so we're just starting to get some of those experiments going now. But what we have been able to do in the meantime is to try and look at least how induction of sort of intracellular HIF signaling in the tumor cells that have disseminated to the bone can also impact dormancy. And we kind of stumbled a little bit backwards into this part of the project. So as I mentioned previously, PTHrP is one of the key factors that are produced by breast cancer cells within that the bone metastatic niche, and that PTHrP really fuels that bone destruction. So there was data from about 20 years ago that was from Jack Martin's group in Australia. And what he had shown was that if you overexpress PTHrP in MCF7 breast cancer cells, so ER-positive breast cancer cells that normally are pretty dormant within the bone marrow, they don't do a whole lot. They will eventually start to grow out in some people's hands. But are relatively dormant in the bone, that if you overexpress PTHrP, these cells become aggressively osteolytic. So fast forward about 15 years, and I was doing some similar experiments with the MCF7 cells where we were knocking down the leukemia inhibitory factor, or LIF receptor, which is part of the interleukin-6 family of cytokines. And we had seen actually a pretty similar phenotype where we saw that the MCF7 cells went from being pretty dormant to being pretty aggressively osteolytic, even though it still wasn't as dramatic as what Jack's group had seen. So I did my first postdoc with Jack Martin. And so at that time, I ended up reaching out to him. And this was, again, purely just from a phenotypic observation, and asked Jack if there was any chance we could get our hands on those PTHrP cells that he had. So it turns out they were still hanging around, and we were able to look in those cells, and we saw that LIF receptor was actually significantly decreased when PTHrP was overexpressed. And we also saw a reduction in one of the LIF receptor downstream targets, which is SOX3. So Jack and I did a little bit more work at that point, and this was after I had left his lab. This was while I was at Vanderbilt, after I'd started at my lab. We did a project where we actually confirmed that the effects of PTHrP on LIF receptor are not through the canonical cyclic AMP signaling that PTHrP usually induces, but it was clearly having an effect on gene expression. And so it suggested that it was all independent of the PTH receptor on breast cancer cells. So what we ended up doing was to start another project with Jack, where his lab had actually designed all of these different PTHrP mutant plasmids, and he sent them to us and very kindly shared them with us. And this is all work that's being done by my student Courtney in the lab right now. So what she has are these plasmids from Jack, where we have either just a control vector, which is MSCB, or we have this full-length secreted vector where where they have just overexpressed the full-length secreted form of PTHrP. They also have a version where they have a mutant version where they've just deleted the nuclear localization sequence. And then they have a mutant where they've truncated the protein, deleted the NLS as well as the C-terminal region. So within the PTHrP protein, there is a secretion signal. There's this PTH receptor activating domain that we would argue probably isn't the most important for breast cancer. A mid region with pretty unknown activity, as well as the nuclear localization signal, and then a C-terminal region, which also contains a nuclear export signal and can also regulate osteoclastogenesis. So what Courtney did was she took all of these plasmids, put them into MCF7 cells, and then she first just validated that we actually see the appropriate proteins. And it's worth noting here as well that the commercial antibodies for PTHrP are pretty garbage. And so we've had to use an HA tag. We have an HA tag on the end of all of these, and that's what we've used for our Western blotting here and then for our chip later. So what she saw, though, was that we did see expression of the proteins. They're quite small, but we saw them roughly around the size that we would have expected. And we also confirmed using qPCR primers to each of the specific domains that we see expression of these, where we would expect for the different plasmids. So Courtney's hypothesis was really, firstly, she wanted to start with the in vivo experiments because that's really what we were working from, was this phenotypic observation, that she thought that deletion of the PTHrP nuclear localization sequence might prevent the tumor cells from actually inducing the osteolysis that Jack's group had previously seen with PTHrP overexpression. And this was mainly due to the fact that previous publication had suggested that the nuclear activity of PTHrP might actually push tumor cells into the cell cycle. So this seemed like a reasonable sort of hypothesis going into it. So what we were pretty surprised to see then was that when we injected these cells into the mice by intracardiac injection and looked at bone destruction, that you see, if you look at the full-length secreted version here, and I'll keep this sort of cartoon up for most of the remaining slides so you can refer back to it. But when you look at full-length secreted PTHrP, we saw a trend towards an increase in bone destruction, certainly not as dramatic as what had been shown way back when, but this was also a slightly different plasmid. And then what we saw was that instead of reversing any kind of bone destruction that was occurring, we actually saw an even further increase in osteolysis. So we saw a really dramatic increase in lesion area and lesion number. And the mice that had the tumor cells expressing cells that lacked the nuclear localization sequence. So that was pretty opposite from what we had anticipated. So we next thought, okay, well then I think we need to, figure out then how are these cells actually even just behaving within the primary tumor site? So how do they grow? Are there differences in proliferation there just at baseline between the cells? So we performed just a orthotopic experiment where we injected these cells into the mammary fat pad. And we looked first at time to tumor palpation. And this became really interesting because if you see the red line here, this is our control tumor cells, which, this was pretty much what we would have expected for the time to palpation. For the full-length secreted, which is in blue, we really didn't see much difference in primary tumor growth. But we did see when you look at the DNLS cells, so these are the cells lacking the nuclear localization sequence, that these actually had a much faster time to tumor palpation. So the tumors seemed to be much more aggressive. And if you look actually at the tumor weights, you'll see, again, no change with the full-length secreted, but we saw an increase with the cells lacking the NLS. But what was really interesting was that if you deleted the NLS as well as the C-terminal region, so all we've done is just to also cut off the C-term, that completely reversed the ability of the cells to grow really quickly and become really aggressive. And you can see this with the time to tumor palpation. They were slower growing, and they also had, they were smaller, much smaller at sacrifice. So we have started staining for just Ki67 within these tumors to try and see if the cells are in fact more proliferative in the DNLS. And Courtney is quantifying all of this right now, but so far based on the data that I've looked at, it does look like there's likely to be an increase in proliferation that we see with the staining in the DNLS cells, so the cells that lack the NLS. And then conversely, it does appear that there is also likely to be a reduction in proliferation in the cells that lack the NLS as well as the C-term. So it does seem to sort of reverse that proliferative effect. So we decided, because we really needed to try and work out more about what's actually happening with the signaling and how are we getting such divergent phenotypes, especially between the primary tumor and the bone metastatic site, that we decided to do RNA-seq on these cell lines. So we just took the cells cultured in normoxia in vitro, and then we did RNA-seq and gene set enrichment analysis on them. And so we had several hundred genes that changed, and we were really interested for the bone. We were really interested in these genes that came up in all three, as well as the ones that came up between the DNLS and the DNLS plus C-term mutants, because all three of the mutants had elevated bone destruction in vivo. So for the bone phenotype, these were really of interest. For the primary tumor phenotype, it was more about what's really different between the three. But for this talk, we've really focused on these cohorts here. So the one of the first things that really jumped out at us that was being expressed between all three was enriched between all three was EMT. So EMT was significantly upregulated across all three mutants. So we decided to look at just a baseline panel of EMT markers within all of the mutants. And we saw that there seems to be really not a huge difference here. If anything, there's maybe a slight reduction in beta-catenin, a little higher vimentin, and a little lower E-cadherin in the DNLS plus C-term mutant. And we also saw, when we looked at some of the markers by qPCR, we did see an enrichment for some of these other EMT markers. So this loss of beta-catenin and E-cadherin and increase in vimentin would be consistent with a potential EMT-like phenotype. And then once we saw these markers as well, that really seemed to only be enriched within that the NLS plus C-term deleted mutant, this suggested to us that maybe these cells might be undergoing EMT. And so this would still potentially fit with the phenotypes that we had seen. So one thing to keep in mind is that when cells undergo EMT, they've also been shown to downregulate proliferation. And so that would fit with the staining that we had seen, where we have a reduction in proliferation, increase in EMT that could potentially slow down the primary tumor growth, but would still make them invasive within the tumor. So it could be that they're still able to efficiently invade within the bone marrow, and that can actually lead to the osteolytic bone destruction. So that's what we think may be happening with those cells. But it still didn't really quite tell us about the DNLS cells. So again, we actually went after this just to look at the bone phenotype, but we ended up coming up with a potential answer for some of those primary tumor mechanisms. So we went back again to the gene set enrichment analysis and looked at overlap again between these to try and figure out what's going on with the DNLS. And one of the factors that really jumped out to us again was that was enriched was hypoxia. And again, obviously we have a lot of interest in hypoxia in the lab, but there's also a lot known about hypoxic regulation of dormant tumor cells. And so what we did was to first just look at whether or not HIF-1 alpha is regulated. And it turns out that if you overexpress PTHrP, you do actually significantly increase HIF-1 alpha transcript levels, but only in the full length secreted, which would be within the nucleus. So that sort of makes sense, but is physiologically probably not important because that's going to get degraded. So we looked at the protein level, and it turns out that even though the DNLS cells, once you exclude the cells from the nucleus, that these cells don't increase transcription, but they do potentially stabilize HIF-1 alpha. So we had a significant increase in the HIF-1 alpha protein levels only in the DNLS cells. And one of the other markers that had, or one of the other pathways that had come up in the gene set enrichment was also glycolysis, specifically within the DNLS cells. And so we collaborated with Dr. Beth Rendina Rudy at Vanderbilt, who ran a seahorse assay for us. And it turns out that the ECAR, or acidification rates, are increased in the DNLS cells, which is suggestive that the cells may be undergoing glycolysis. And so our working hypothesis for this is that we think that PTHrP acting outside of the nucleus can potentially stabilize HIF-1, induce glycolysis, and that this just ramps up the tumor cells so that they're really metabolically active, and that this can then potentially lead to that exit from dormancy. So just to sort of summarize this, our idea here then is that the mechanism potentially for our primary tumor and for our sort of our rapid growth and accelerated growth with the DNLS cells and their bone destruction is due to them just being amped up from the glycolytic and metabolic changes. So the DNLS plus C term, though, deletion of just that C term region seems to switch this so that's no longer increase in proliferation. We now have this reduction in proliferation, but we have this potential increase in EMT that slows down the primary tumor growth, but may help the tumor cells invade the bone better. And again, you know, all of this is a snapshot, so there could still be changes in proliferation that we're just not capturing right this minute. So for just the last slide or two, we've also continued to look further at how PTHrP and HIF and LIF receptor are all sort of acting together to regulate dormancy. So I showed several slides back that PTHrP had inhibited LIF receptor sort of at that transcription level. We've also confirmed at the protein level that LIF receptor is downregulated in all 3 of the PTHrP overexpression mutants. So that sort of holds true and is consistent with the increased bone destruction that's observed in those mutants as well. Then we have also then seen that PTHrP again might stabilize HIF1 alpha. And so one question we had was, is it possible that PTHrP regulates HIF1 and that this regulates LIF receptor? And it turns out we sort of already knew the answer, because we had previously shown that LIF receptor is actually negatively regulated by hypoxia, but it's independent of HIF1 alpha. So we think PTHrP independently regulates HIF1 alpha and LIF receptor, and these are 2 distinct mechanisms to promote exit from dormancy. And so we've pursued this a little further to see how PTHrP might regulate LIF receptor. And to do this, we did CHIP to see if there was any chance that it's actually binding to the LIF receptor promoter. And it turns out that if we perform CHIP using an HA targeted antibody for PTHrP, that we do see enrichment within, this is all the proximal promoter of LIF receptor, that we do see enrichment within this particular site of the promoter. So this is site 3. And this is interesting because, one, no one's ever shown PTHrP actually binding to anything in the nucleus. And it's quite possible that it's really just part of a complex and not directly binding the DNA. There is no known DNA binding domain for PTHrP, so it's likely part of a complex. But we've noticed even within this part of the LIF receptor promoter that this also seems to be the region where LIF receptor is really regulated by hypoxia as well. So everything seems to be working together to kind of to push the cells out of dormancy. So in hypoxia, you'll see in the same part of the promoter in this region 3, if you look at just histone H3 lysine 9, the methylation, which is a repressive mark for this histone, is actually increased within the LIF receptor promoter, and acetylation is reduced, so suggesting that you have a repression of LIF receptor transcription. And we also see that this particular region of the LIF receptor promoter is where we can even target LIF receptor and increase its expression using histone deacetylase inhibitors. So really trying to go after the acetylation that seems to regulate LIF receptor. And so we can really stimulate LIF receptor quite a bit by using those HDAC inhibitors. So this is my last slide, just to sort of sum up everything. So we've already covered all of the mechanisms that we think are happening, or at least the phenomena that we've observed with HIF signaling regulation to these distant sites. And this was, again, sort of summarizing the last bit of our data from the primary tumor, where we think that this truncated form of PTHrP is really regulating EMT and inhibiting proliferation, and that adding back in that C-terminal region actually pushes towards a proliferative phenotype. But what we think is happening within the bone itself is that you almost certainly have normoxic and hypoxic tumors, and we see that at the histological level. So we think in the hypoxic tumor cells that are there, that one of the ways that the cells are exiting dormancy is through this LIF receptor repression, through changes to the chromatin and the histones. But in normoxic tumor cells that are there, we think that PTHrP may actually be switching on hypoxia signaling through HIF1. and that it might also be repressing LIF receptor as well to also enable the tumor cells to exit dormancy. And so we think there's quite a few mechanisms that are going on, but there's definitely a lot of interplay and crosstalk between PTHrP and HIF and LIF receptor, we think, particularly within the bone marrow. So with that, I would just like to thank all of the people and everyone in my lab who has been working so hard on these projects. So this was our holiday, socially distanced holiday party where we did a remote escape game. And Vera and Courtney are really the ones who have done so much work on this. Tony also did a lot of the mouse work for the genetic models as well. And then Miranda, who's not pictured here, who is my former student, who's now a postdoc at NCI, she did all the work with the HDAC inhibitors and has that's a whole extra story as well. So I'd just like to acknowledge our funding sources, all of our collaborators who have been really fantastic, and I'm happy to take any questions. And I will also stop sharing, so hopefully that will be better for the sound.
Great, thank you, Dr. Johnson. It was an excellent talk. Looks like, while people are starting to type in, Phil already has a question. Can you comment about the unique impact of blood and lymphatics in response to hypoxia in the bone tumor microenvironment?
Yeah, hold on just a second. I'm having trouble hearing through the phone, so I'm gonna switch over the
Oh, sure.
Sorry, can you hear me okay now that it's not shared?
Yeah, much better. The joys of technology. So
hold on, sorry, it's still, hang on, it's still going to send all the audio through there. I'm gonna leave there. Okay.
All right, can you hear me now?
I can't hear you.
What about now?
Hear me clear?
Yeah, you're cutting out a little bit again, but we can figure it out.
Okay. Sorry, I'm not sure what's going on with it.
That's okay.